Hi everybody, my name is Alicia Martinez-Varela and I am a PhD student at the Environmental Assessment and Water Research Institute from CSIC in Barcelona, and this is an oral communication for the 7th International Symposium on Marine Sciences titled Large Enrichment of Anthropogenic Organic Matter Degrading Bacteria in the Sea Surface Microlayer at Coastal Antarctica. Let's start with a brief introduction on what is the sea surface microlayer. It is the uppermost top layer of the marine water column and constitutes the boundary layer where gas and particle exchange between the sea and the atmosphere occurs. It is a microenvironment because it is not larger than 1 millimeter. However, it does exhibit distinct and extreme and very variable physical, chemical, and biological features when you compare it to the underlying blue water. And from now on, we will call the underlying blue water SSL, which is subsurface layer. The structure and function of the SML bacterial residence, known as Bacterial Livingston, has been poorly characterized, probably due to the difficulties when sampling this layer. However, Bacterial Livingston is thought to play a central role in the mediation of many air-sea exchange interactions and biogeochemical and climate-related processes at both regional but also a global scale. Far from being a benevolent habitat for microbial life, Bacterial Livingston withstand intense environmental stresses such as harsh ultraviolet radiation, wind speed turbulences, and also enrichment of toxic and persistent compounds such as heavy metals or hydrophobic organic pollutants. Also, besides this, there is also an enrichment of organic matter with surfactant properties, both from anthropogenic and biogenic source. The accumulation of this pool of organic matter can promote bacterial growth. The SML is also a direct recipient of atmospherically deposited aerosols, and the diffusive atmosphere-ocean exchange and snow deposition are the main sources, but not only of semi-volatile organic pollutants, but also other hydrophobic and surfactant-like organic pollutants such as perfluoroalkyl substances. All these OPs become part of the anthropogenic dissolved organic carbon pool, which due to its hydrophobic and surfactant-like properties is enriched in the SML, as reported in several studies. And the accumulation of other surfactant-like and organic matter is driven by not only deposition of atmospheric compounds, but also driven by rising bubbles or other compounds attached to buoyant particles. Also, the removal processes of these compounds from the SML is there is volatilization, cis-spray aerolization, photodegradation, settling of particulate matter, and also microbial degradation. And that's because the ocean microbiome harbors a subset of bacteria known to play a central role in the biodegradation of adduct. Acute pollution events especially have served to reveal the genetic battery required for degradation of these compounds, and also which are the taxa and what are the degradation pathways. All these processes depict the SML as a very dynamic system between the interactions of organic matter and bacterial Livingston. However, it has not been studied in depth yet and never addressed in the Southern Ocean, where we find that all these semi-volatile adduct that can be transported atmospherically or by current very long distances arrive to Antarctica. So this is the object of our study. And the goals are to better understand the temporal dynamics and main environmental drivers of bacterial abundance and community composition in the SML in the maritime Antarctica. And we did this by comparing the SML and SSL communities' taxonomical structure during the nutrient-rich upwelling summer season in South Bay, measuring not only the bacterial abundance and community composition and analyzing for both layers, but also correlating these data with meteorological conditions and physical and chemical variables from water and air. And also we use perfluoroalkyl substances as surrogates of hydrophobic and surfactant organic matter, as well as the SML stability. Results and discussion section. First of all, I'm going to characterize the sites. We sampled at 5 different stations over the austral summer of 2018 in the South Shetland Island, Livingston Island, the South Shetland Archipelago in Antarctica, where the Spanish Antarctic base is located. We sampled with a glass plate SML sampler, first described by Harvey and Bacterial in 1972. And During summer months, strong upwelling events are caused by the Antarctic divergence and westerly winds, inducing nutrient-rich waters. And in this plot, you can see the three first plots are the measurements for inorganic nutrients. In orange, you can see the SML measurements, and in blue, the SSL measurements. And these confirmed that we were working on a non-limited nutrient system. Also, the last plot, as we mentioned earlier, the level of pollution of anthropogenic dissolved organic carbon was tracked by taking the concentrations of perfluoroalkyl substances, from now on PFAS, as proxy of ADOC. These are synthetic compounds globally distributed by oceanic currents and by atmospheric transport through marine aerosols. And PFAS here were measured for the same set of samples than Bacterial-Newton, and will be soon published by Casas et al. And thanks to the ambifilling nature of these compounds, both hydrophobic and surfactant, these accumulate at the SML. And as surfactants have already been used as surrogates of the occurrence of the SML and its stability, that's the reason why we took these as a proxy of developed SML-enriched in ADOC. In particularly, here we used the perfluorooctanosulfonic acid, PFOS, from now on, as it is easier to pronounce. And this compound was found enriched from 1.2 to 26.3, enriched at the SML over our timeline. And also, for example, we see here a peak over this period of time with a great uncoupling between both layers, so indicating a very high stable event at the SML over this period. And contrasting with these conditions, we see minor difference between the PFOS between both layers at these gray marked days. Consistent with disruption of the SML, possibly caused by wind speed that you have here. The darker the blue, the faster was the wind. So when it comes to structure of Bacterial-Newton and bacterioplankton, we observed a significant higher bacterial abundance of Bacterial-Newton compared to bacterioplankton. Consistent with observations in other oceanic regions. And when it comes to the bacterial assemblages, we observed that both layers shared dominant groups, which were Flavobacteria, Gammaproteobacteria, SAR11, and Rhodobacteriales, and also that both microbiomes included SML and SSL microbiomes included genera with non-facultative and obligate ADOC degrading strains. And when we looked at a finer taxonomical resolution, we found that higher relative abundance of specific ADOC degrading bacteria known to be stimulated under high ADOC concentrations were observed, and the fold change of these groups in the SML versus the SSL range from 1.5 to 50. So when we look specifically, on average, taking all the data set, significant higher relative abundance were observed for Gammaproteobacteria ADOC degrading strains and Flavobacteria ADOC degrading strains. And also we observed fold increase larger than 2 for these ADOC associated groups, Alteromonadales, Sphingomonadales, and Caulobacteriales. All of them enriched at the SML in general across all our data set. Also, when we look in a day-by-day basis, we saw Gammaproteobacterial groups were systematically highly enriched at the SML, positive fold changes in red and negative fold changes in blue. And in particular, genera Colvelia, Pseudoalteromonas, and Pseudomonas, which are generalist strains with rapid facultative degrading capabilities towards ADOC, but were all generally enriched and bloomed from the 24th of January onwards. And all of these are also prone to degrade other dissolved organic matter. From here, as some of these stacks are not obligate ADOC consumers, it is relevant to address whether SML is enriched in dissolved organic carbon, DOC, or only ADOC and biosurfactants. Previous work have shown that DOC in seawater from this region had values around 62 micromole carbon per liter, while the DOC at the SML was 69 micromole carbon per liter. So the difference in concentrations are small in terms of bulk DOC. And in Antarctica, several works have reported the enrichment of hydrophobic pollutants in the Southern Ocean, such as alkanes, PCBs, PHs, PFAS, and others. So this suggests that the moderate reported enrichment of DOC in the SML, when occurring, may be mostly driven by the fractions of hydrophobic and surfactant-like DOC, but probably not other pools of DOC. What happens with environmental drivers of community composition? We did varied correlations between environmental and bacterial composition variables, considering all measures in the SML and SSL. And we saw that nutrients pointed to be a pivotal role as key drivers of microbial, having a pivotal role, of key drivers of microbial composition. But also we saw that relative abundances of Alteromonadales and Gammaproteobacteria ad hoc degrading strains were positively correlated to concentrations of PFAS families, in particular PFOS and other PFAS families. And at the same time, perfluorohexane carboxylic acid correlated negatively to dominant Rhodobacterales and alpha Protobacteria ad hoc degrading strains. So from here, we have two strategies which are consistent with ad hoc modulating the structure of community composition and aligning at the same time with this dual effect of ad hoc compounds on marine microorganisms. From one side, we have ad hoc can be a source of nutrient and organic carbon source. But oppositely to this strategy, other compounds can also induce toxic effects to bacteria. So here are our conclusions. We found that total bacterial abundances were higher in the SML than in the SSL. Also, while dominant groups were shared between both layers, we found difference with higher relative abundance at the SML of taxa reported to be ad hoc degraders, such as Gammaproteobacteria ad hoc degrading strains, Flavobacteria ad hoc degrading strains, and other orders associated to ad hoc. Also, we found significant positive correlations between the relative abundance of Alteromonadales, Gammaprotobacteria ad hoc degrading strains, and Sphingomonadales, and the concentrations of PFAS used as proxy of hydrophobic and surfactant-like ad hoc. So from here, our findings provide experimental evidence that ad hoc accumulation at the SML could be another, although neglected until today, community composition driver. And finally, dual effect. Ad hoc can become potential growth substrate for microorganisms, but also act as a toxic agent and hamper bacterial growth of other taxa. So thank you very much for listening. Here I present you our research group, Global Change in Genomics Biogeochemistry Research Group, led by Jordi Dags and Maria Villacosta. It's all about teamwork in the end. And thank you, special thanks to the organization for pushing forward and adapting the symposia to the new circumstances. Thanks for your attention, of course, and I hope to see each other soon face to face.